Hello future doctors, how are you all? Welcome back to MedSteps YouTube channel. So right now we are doing first aid series. So in microbiology we are discussing about properties of growth media. In our last video we already discussed about stains, how we have to do the stains and what purpose we are using the gram stain and chocolate agar stain. These are the things we already discussed. So today, not all bacteria can grow anywhere. So some need like special food, or some bacteria or organisms need some protection, and some need special conditions. That's why we use different culture medias. So here if you are seeing in this slides, you can see here the same type of media can possess both or neither of these properties. So first of all, selective media. So what is selective media? Selective media means selective media allow the growth of a particular organisms and while inhibiting the growth of others. That means it will prevent the growth of other organisms. So why it's important means the selective media will help to inhibit the organisms. So based on that we can easily find out which antibiotic is sensitive. So based on that we can use that antibiotic to kill that bacteria. Okay. So antibiotics are chemicals are we are going to add, or whenever we are adding it will suppress the unwanted organisms. Only the target organisms will become survive. Okay. So here in example you can see Thayer-Martin agar. So this Thayer-Martin agar, it contains antibiotics. It will used for Neisseria gonorrhea or Neisseria meningitis. So what does this Thayer agar contains means, like components like vancomycin for killing gram positive bacteria and colistin for killing gram negative bacteria, or trimethoprim for prevents protease, and nystatin for killing fungal type of infections or organisms. Only Neisseria will grow in Thayer-Martin contents. So this Thayer-Martin agar contains antibiotic that allow the selective growth of Neisseria by inhibiting the growth of other sensitive organisms. So for this thing we have one mnemonic. That mnemonic we call very typical culture Neisseria. If you are seeing here, this is the Neisseria gonorrhea or Neisseria meningitis. We are using that media we call Thayer-Martin agar. It's like for selective favor growth of Neisseria by inhibiting growth of gram positive organisms with vancomycin and gram negative organisms except like Neisseria with trimethoprim, colistin and fungi with nystatin. So the mnemonic is very typically cultures Neisseria. Okay. So next one you can see here, indicator differential media. So whenever any patient coming with any infection or microorganisms, for finding the thing we have to do culture sensitivity or specificity. So here in differential media it differentiate organisms based on biochemical reactions, usually by their color, that organisms color change. So we have one color word, that mnemonic, that is color change. Okay. So in this indicator media we can see it contains sugar, example like lactose, or pH indicator. These organisms metabolize sugar differently. Okay. So in examples we can see like MacConkey agar. This MacConkey agar contains pH indicator and lactulose fermenter, like E. coli will convert lactose to acidic metabolites. So that's why in this agar we can see color changes. So lactose, the main function is it's a sugar source. pH indicator, it will detect the acid, and bile salts, it inhibits gram positive. So how we can do interpretation means, here whenever patient coming with this type of indicator differential media, we can see some organisms. The lactose fermenter, like example like E. coli, we can see pink colonies. In non-lactose fermenters like salmonella, we can see colorless. So MacConkey is one type of media or agar contains is selective for differentials. So, next media is enrichment media. This enrichment media provides special growth factors required by fastidious organisms, like it will provide some fastidious organisms, some bacteria like lack of enzymes, they require external growth factors. So, you can see here we have some examples. In that examples you can see like Haemophilus influenza. So, it contains factor 10 like hematin and factor 5 NAD+. This is blood is heated. Whenever we are heating that blood, the red blood cells will become breakdown, then these factors will release. So, that is why the enrichment media we can use chocolate agar to find out these factors. Okay, then only we can like diagnose that patient have Haemophilus influenza. Okay, or the anaerobic conditions needed by like Clostridium species. And next one, this Clostridium species, it's like it requires absence of oxygen. So, at last we can compare from like selective media and what are the things it is doing. The selective media will inhibit others, the differentials will do color change, the indicator differential media. The enrichment, it will provide the nutrition. Okay. So, here we have some special culture requirements, that is Haemophilus influenza. The media will be like chocolate agar, it requires factor 5 and NAD+ and factor 10 hematin. Next one, Neisseria gonorrhea. In this Neisseria gonorrhea and Neisseria meningitis, the media will be like Thayer-Martin agar, the property will be like selective one. Next one, Bordetella pertussis. The media will be like Bordet, Gango agar, and another one Regan Lowe medium. It contains potato extract, charcoal, blood and antibiotics. Next one you can see Corynebacterium diphtheria, and the media will be like Tellurite agar and Loeffler medium. So, next we can see here, that is Mycobacterium tuberculosis. The media will be like Lowenstein and Jensen agar, it contains malachite, green, and it inhibits contaminants. Next one you can see Mycoplasma pneumoniae. The agar will be like Eaton agar, it requires cholesterol. Next one you can see here, why it requires cholesterol, because no cell wall in Mycoplasma pneumoniae. Next we can see lactose fermenting enterics. The media will be like MacConkey agar, and it ferments produces acid, and it causes colonies to turn into pink in color. So, next one you can see E. coli. That E. coli we have one media, that is EMB, that means eosin methylene blue agar, that colonies with green metallic sheen. So, next one we can see here, that is Legionella. The Legionella, the media will be like charcoal, yeast extract agar, it buffered with cysteine and iron. So, it does not grow on routine media, that is why that for Legionella charcoal yeast extract agar is very important, because it is buffering with cysteine and iron. Next one, fungi. The fungi is like that media will be like Saborad agar. That mnemonic we have, that is SABS, a fun guy. So, the selective media kills others, the differential media will change color of the microorganisms, the enrichment media will feed bacteria, and special media will solve our exam questions. Okay. So, these are the things. Whenever we are thinking about any microbiology tips or anything, that time we have for everything we have some mnemonics. So, we have to write and we have to remember, then only we can, in exam time we will get easy. Okay. So, if you didn't subscribe my channel, like, share and subscribe. We will meet in our next video. Till the time, take care. Bye bye.